Okay, I'm going to go over the lab final review. Just as a reminder, if you've been to lab, you've done your lab prep work, which was required, and you've been conscious and alert and doing your part in your lab group, none of these questions should be surprises. I'm not saying you'll remember everything, but that's how I designed the lab final, is that can you actually do you actually understand what we've covered and what's been important. So some labs are going to be way more important than others, and that's why you'll see me highlight them here. So the final review is just to get you to, I'm going to really expand on what we have here because it's just getting you thinking about what was important in a microbiology lab. All right, let's jump in. So basic laboratory safety. What is the most important thing to do to keep yourself safe in the microbiology lab? To come prepared. Now I know having your hair tied back and wearing the right shoes and not being careful with Bunsen burners, all those are important. Well, that comes with preparation. So to me, mistakes happen when you don't know what you're doing, of course. And if you're confused about what the next step is and you're handling materials while looking at what the next step is, that's just a recipe for disaster. So when I say come prepared, if you know, that's why even though I have you guys come prepared to lab, we still go through every step before you get started, because being confident that you know what you're doing and what you should be doing next is going to keep you safe. So you'll understand, like, I should have gloves on for this. I've got to be careful with the Bunsen burner for that. So all those minor details fall under this umbrella. Coming prepared is the most important thing to do in the lab. I care much more about you guys and your safety than I do any individual lab. So that's why I need you to take safety seriously. So, all right, next, aseptic techniques. Why is aseptic technique important in the micro lab? So first, what does that mean? Asepsis is not sterile. We can't create a sterile environment, but aseptic technique is basically a lack of significant contamination. So we can do everything perfectly and have contamination. We saw that happen this semester sometimes. But so why is it important in the micro lab? Because if we're trying, we clearly don't want to spread microbes unnecessarily. So that's obviously important. But we also we want to make sure that we're working with pure cultures. If you want to see if E. coli can be killed by antibiotic X, then we have to make sure it's E. coli. And if we don't follow the right techniques, we have contamination, and that's a problem. So Robert Cook developed these techniques using slices of potatoes and things like that because he couldn't use his own postulates to say that this organism caused this disease unless he could isolate pure cultures. So it really ties into the next point too. What three things does aseptic technique protect in the lab? That would be you. I mean, my priority is keeping you safe. Number two would be your experiment. Making sure that you're keeping your microbes from contaminating the environment, but also other microbes from contaminating your experiment. All makes sense. And number three is the lab, the location. So you've got your experiment and the location. Obviously, the location would be protecting your fellow classmates and protecting the other people that use the room. So those are the three things that helps protect in the lab. Next, we have microscopes. Let me scroll down a bit. Why are our microscopes called compound microscopes? So our microscopes are actually called binocular parfocal compound light microscopes. Binocular, just two eyepieces. Parfocal, that's actually good news. It means that these objectives are linked together in a way that if you get them focused well at the lower magnifications, it'll make your job exponentially easier as you go. Older microscopes, you had to basically refocus at each move of each objective just got you in the right ballpark for the next objective. So binocular, parfocal, they're called compound light microscopes because a simple microscope would have one lens. Compound microscopes use multiple lenses, and that's not the four objective. That's not the scanning power, low power, high dry power, and oil immersion power. It's whatever objective you're using plus the lens that's in the eyepiece in the oculars. That's why it's called a compound light microscope. Called a light microscope because it uses light, compared to microscopes that use lasers and beams of electrons, etc. All right, how many different objectives are there on our microscope? So we have the scanning power, which was a 4x objective. And remember that you multiply that by 10 because there's a 10x objective in the eyepiece. So a total magnification of 40. Then we had low power. So older microscopes didn't have scanning power. So low power is 10x for a total magnification of 100, 10 times 10. Then we have the high dry power, which is the highest power we can get without magnification. Or magnification without using immersion oil, sorry. So that's why it's called high dry power. 40x for a total magnification of 400. And then we have the oil immersion microscopes, which have 100x magnification for a total magnification of 1,000. So that answers the two questions. How many different objectives are there? There's the 4. And then what's the highest total magnification we can achieve? That is 1,000x. There are some light microscopes that can get to 2,500x. We don't have any of them. They have the normal oil immersion objective, but they have a 25x eyepieces. So I'm trying to get the money together for one of those. Next, smear preparation and staining. That was a 2-week lab we did. So what is a smear? It's a very thin preparation of microbes on a slide. So what surface do you make smears on? You were streaking or spreading microbes on a slide. And then should they be thick or thin? The answer is thin. And why? You have to be able to see through them. If you have too many microbes there, you're going to see a lot of color when you do your staining, but you won't be able to see individual organisms. So just to review with smear prep, remember we transferred 2 loops of organisms from broth onto a slide. We spread it real nice and thin. And then we waited, because the key step there is it has to air dry. If you try to heat fix these smears before they're air dried, it's going to have a very negative effect. It's going to damage the cells. They may not even hold stain, or they definitely won't look right. So you add your organism, spread it thin, air dry it, and then heat fix it. And that heat fixing is very important because it actually attaches the organisms to the slide. You start pouring water and acid and alcohol and stain all over these slides. If they're not properly heat fixed, the bacteria will be gone. All right, next is staining. So what are the 3 staining techniques that we completed in the lab? I'm just going to scroll down a bit here. We talked about things like negative staining and flagella staining and simple staining. But the 3 stain techniques that we used we did the gram stain, the acid-fast stain, and the spore stain. So, I was going to answer the next few questions. We'll just do them. Which is generally considered the most important staining technique to start with? That is the gram stain. So because however many bacteria there are, some would say 1 million, some say 4 million. We have no idea because so many can't be grown on petri dishes. However many bacteria there are, basically we'll just say they're half and half. So the gram stain points you in the right direction. Are you dealing with a gram-positive bacteria or a gram-negative? So if we have a sample of staph and a sample of E. coli, the gram stain will point us at those 2 different paths, because staph is gram-positive and E. coli is gram-negative. So the gram stain is definitely the place to start. There are only a few organisms where you're not worth starting with the gram stain first. All right, what kind of bacteria produce spores? That can be seen with a spore stain. So the spore stain looks for the 2 genus Clostridium and Bacillus. So Bacillus, a lot we work with Bacillus subtilis, which actually is it makes some of your fermented foods, and it's a soil organism. But there you have Bacillus anthrax. Clostridium, you've got Clostridium tetanus, C. diff, Clostridium botulinum, etc. So the spore stain is going to help us find the Clostridium and the Bacillus. Next, what kind of bacteria would be acid-fast positive? So the acid-fast stain is primarily used for the Mycobacterium, like Mycobacterium tuberculosis, which causes tuberculosis, and Mycobacterium leprae, which causes leprosy. But the acid-fast stain can also find the nocardia. That's a relatively minor deal, but you should still know it. All right, so the acid-fast positive organisms would be those, the Mycobacterium and nocardia. I will not ask you to know the steps of the acid-fast and spore stain. I want you to know when we would use them in the big picture stuff. But the gram stain is very important, so we will go through the key steps. I'm not saying how many seconds you use water, that kind of stuff. But know the key steps of the gram stain technique. What step actually determines whether bacteria will stain as gram-positive or gram-negative? So we start with crystal violet, our purple, which stains everything purple. And then we go to the iodine. And when crystal violet, which is small, and iodine, which is small, form a complex together, it makes them bigger. So it's the mordant, which helps hold the stain in. So at this point, all the cells, whether they're gram-positive or gram-negative, they're all stained purple, and that purple is locked into those cells at this point. But then the next step, which is the step that actually determines which direction you're going to go, is going to be the decolorizing step. So Graham's decolorizer, what it does is the alcohol in the decolorizer actually shrink wraps the gram-positive bacteria, holding the purple in. So it forces purple to stay purple. The alcohol in the Graham's decolorizer pops the lipid outer membrane of a gram-negative bacteria. Let me get a copy. Got a little cough. So the alcohol in the Graham decolorizer pops that outer membrane of a gram-negative, And then since they have a very thin, weak peptidoglycan cell wall to begin with, the purple will ooze out. So after this step, the gram-positives are still purple. The gram-negatives are now colorless. And then the last step is a counter stain, which is safranin, which is red, and it stains everything red, but you can't stain things that are purple red. So it's just a counter stain to highlight the now colorless gram-negative bacteria. But bacteria are hard enough to find if they're not colored. If they're colorless, it's much more difficult. Okay, so that's your gram stain. All right, next, growing bacteria. So you see, I have three sections here. We went through labs on growing, isolating, and then killing bacteria. Growing bacteria. What are the basic ingredients of the nutrient broth we use to feed bacteria in the lab? Is it similar to what humans eat or wildly different? It's actually very, very similar. I mean, they love eating us, so you are what you eat kind of thing. But it's basically pre-digested beef and yeast. Think about, kind of like beef bouillon with some pre-digested protein in it. Got some electrolytes and all that. So it's just basic. They have the same requirements we do. They need a carbon source, they need energy, and that's what it is. So nutrient broth is bacterial food. Think about like dog food. Could you survive on dog food? Sure, we could survive on this too. So it's primarily, I think, pre-digested yeast and beef with some electrolytes added to it and that kind of thing. So it's very similar to what you would eat. Now, not near as tasty, but very similar. But I gotta get these typos fixed. What is agar? So agar. What is it made of? I don't know what it's made of, but what it's made of is a seaweed extract. And why do we use it? So it's very important you understand that agar is not food. It is a thickening agent that creates a growth surface. So agar is a seaweed extract. It's a form of polysaccharide fiber that most bacteria don't eat. So the seaweed digesting bacteria would, of course, but so when we spread bacteria on or in agar plates, what it does is it creates a growth surface. That way, because with a broth, the problem is they can't form colonies. If you want a bacteria to form colonies, you got to put them on a growth surface. So we would have used petri dishes and slants that had agar in it. So it's a thickening agent made of seaweed, and that's why we use it, to create a growth surface. All right, what is the optimum growth temperature of the majority of the bacteria work within the lab? So if you remember from the lab, we used different numbers at different times. So we primarily had a 37-degree incubator, a 30-degree incubator, and a 25-degree incubator. So the most bacteria we cared about are 37 degrees, like the antimicrobic sensitivity testing lab, all four of them 37 degrees. Staph, E. coli, Proteus vulgaris, and Pseudomonas. So we use the 30-degree incubator for most of your soil organisms, like not Bacillus megaterium, but the other Bacillus organism. And 25 degrees we used really just for Serratia marquescens. It can grow at higher temperatures, but if you grow Serratia at 37 degrees, it'll grow fine, but it'll be white. If you grow it at 25, it'll be that red color that we used. So, all right, so that's the optimal growth temperature, 37 degrees Celsius. Remember why? It is because 37 degrees Celsius is 98.6 degrees Fahrenheit. So that's why it's so important. I mean, we are their hosts, so of course they like our core body temperature. All right, do most of the bacteria we care about grow in a neutral pH or one that is highly acidic? The ones we care about are more around neutral. So think, 6, 7 in that area. Highly acidic environments actually inhibit the growth of most pathogens. That's why fermentation was the primary method of food preservation before the refrigerator. If you turned cabbage into sauerkraut, that low pH would keep it from being eaten by microbes. So more closer to a neutral pH. And then your molds and yeast like a little bit slightly more acidic pH. All right, isolating bacteria. What is the goal of the streak plate or surface streaking method that we used in several labs? What are the major difficulties in this technique? So remember the streak plate, I taught you how to use 5 streaks, 4 streaks, 3 streaks, and spread through the middle, or I showed you different methods you could use from the lab manual too. The job is to isolate bacteria. If you do a good job and you see isolated colonies, that means when you did your streak plate method, you spread it so thin, you left a trail of individual bacteria that then formed those colonies of millions or more organisms. So the major difficulty is it's hard to do. It only takes a few minutes and it's cheap, but it's hard to get the streaks perfect. And that's why we practice it half a dozen times. We tried different methods of should you sterilize the loop between every step, every other step. I had you try different techniques because you got to figure out the best way to do this. So it's not easy to spread bacteria so thin, you're spreading individual organisms. So that's the difficulty. All right, what are the advantages to using the pour plate method, which is called the serial or loop dilution method? And what are the disadvantages? That's where we did the lab with the Roman numeral 1, 2, and 3, where you made your own petri dishes. We put organisms inside the agar while it was still molten. We didn't wait and grow it on the surface. So we grew bacteria inside the agar. So then we did a dilution. We put a single loop into 1, and then we grabbed a loop from 1 and put it into 2, and then from 2 to 3. So the major advantage is it's almost guaranteed to work. I think for every group, but maybe one across the entire semester, this semester of doing this, they got a really good looking plate with isolated colonies interspersed throughout the agar. So that's the advantage, it's effective. Disadvantage, time consuming. It takes an hour instead of 5 minutes. And it's wasteful. You're actually making 3 petri dishes knowing 2 of them aren't going to work or aren't going to be what we need. So it's wasteful, so it costs more resources and way more time consuming. But the advantage is it's almost guaranteed to work. And I would never say that about the streak plate method. So I've been doing this for 11 years, and I still have plates that end up going in the garbage with the streak plate method. Okay, killing bacteria and biotechnology. So what disinfectant did we say has been undefeated at killing bacteria in the lab for the last 9 years? That's hydrogen peroxide. Good 20% bleach solution, still very effective, but clearly the best one every semester that I've taught this class, and talking to Rod, who was taught it with 27 years, I think, it's hydrogen peroxide. So we had a case where we tested the hydrogen peroxide solution that they were using in the wellness center, and it didn't work. And I promise you, it's not the hydrogen peroxide's fault. It's either water in a bottle that says it's have hydrogen peroxide, or it's severely diluted. So hydrogen peroxide has been the clear winner. We've had some hand sanitizers work, some don't. Some concentrations of bleach work, some don't. There's here or there. There's some of the disinfectants around the college work, some don't. But hydrogen peroxide H2O2 is what we say is undefeated. All right. Why did you need to use the data table from your lab manual to determine whether or not an antibiotic was effective at killing the bacteria you tested? So that's because we were doing a standardized thing. We used special agar called Mueller-Hinton agar, double thickness plates. And when we spread the bacteria throughout and then put the antibiotic discs on the petri dish, and then saw what sort of zone of inhibition they had, just having a zone around it didn't actually mean anything. Is that if the zone was only 5 millimeters, that way, and you looked at the data table, you might say, well, that means it's resistant, it's not working. If it was 10 millimeters, you might look at the data table and say it's intermediate, sort of works. And at 15 or 17 millimeters, it would be that it's sensitive and works. So just having a zone of inhibition is not enough. How big it is and measuring it, so counting how many millimeters that zone of inhibition was across, and looking at the data table, which based on the antibiotic and the bacteria you're using, that tells you if something is effective. So just seeing a zone means nothing. Some antibiotics need a zone of inhibition of 30, 32. Others need a zone of inhibition of 10 to be sensitive. So that's why the data table and the fact that we standardize it is so important. All right, next. What machine do we use to sterilize the materials we use during labs? Hint, you put your waste in orange blank bags. That is the autoclave. The autoclave uses pressurized steam. It actually can rehydrate spores and then kill them. So it kills everything that we need it to. Our autoclave wouldn't kill prions, infectious proteins, but it kills everything we need it to if we use it at 15 psi, pounds per square inch, at 121 degrees Celsius. So that pressure actually makes it like a pressure cooker, so that water doesn't boil at 100 degrees. It turns into steam at 121. And you do that for 15 minutes. 15 psi, 15 minutes, 121 degrees. I actually run it. I shouldn't even tell you, but I run it at 20 minutes. But just better safe than sorry. But 15 minutes is what I want you to know. Okay, and then when do you disinfect your lab benches? Before we start, after we stop, and anytime there's a spill. So that's to protect you, your group mates, your classmates, and whoever else uses the lab. All right, biotechnology. Why did we work with 2 tubes of bacteria during our P-Glow lab, even though we only genetically transformed 1 of them? Well, the second tube was our control tube. I like to call a control a standard of comparison. So we treated that tube, the negative tube, the red one, exactly the same as the positive tube, the green one, except for one step. We added transformation solution to both. We added broth to both later. We heat shocked both. The only step we did differently is we added the plasmid, the peak glow plasmid, to the positive tube and not the negative tube. So what we did is we created a better experiment because there was only one variable that was different. That's the best science you can do. So we actually got to see how normal, non-genetically modified E. coli would behave in all the situations we put it in. And then we got to compare that to our genetically transformed ones. All right, why did we not have to worry about isolating the bacteria on the Petri dishes? What did the isolation for us? So if you remember during that peak glow lab, you spread the bacteria thin throughout the entire, you covered the whole plate in bacteria. That's not going to leave isolated colonies, but you ended up with isolated colonies. So the medium we use is an example of a selective media. And that's because, so you didn't have to do a streak method. You didn't have to thin out these bacteria. What was going to do it was the ampicillin, the antibiotic. We taught our genetically modified bacteria to resist the antibiotic ampicillin. Then we coated the inside and out, the petri dish with it. So the only bacteria that survived were ones that could resist ampicillin. That's why you didn't have to spread it thin, and that's why it's the best example in lab of what we used called a selective media, where the growth surface actually helped you determine what organisms were there, because we knew if they were still there, that they had to be able to resist ampicillin, the antibiotic. Okay, that should help you guys crush this lab final. Go for it.